which is gonna be cellular biology. Mr. Ling, do you want to explain what this week's gonna be and what is all this that we're doing?
All right, so behind me now, I have a cell. So cellular biologists study cells. So some of you guess things are really big, some of you guess things are really small. Cells are kind of in between. They're really small, but they're not as small as an atom. And these cells are the building blocks of life. So everything that is alive is made up of cells, including me. I'm made up of lots of little cells that make up my whole body. I'm dressed for Halloween as a skunk. You see my tail? Skunks are made of cells too. Everything that's alive is made of cells. Bacteria, protists, mushrooms, plants, they're all made of these little building blocks called cells. And cellular biologists work with these cells from all different living things, all different organisms to study how they work.
Do you guys know what DNA could be? Any guesses for what DNA is by chance?
It's the blood.
The blood, that's really close. All our blood has these cells with all DNA. So, what does DNA exactly work for in our body? Does anyone else have a guess?
So the DNA is a recipe for your body. Oh.
That's a really great guess. A recipe for your whole body. That's really great. And that's an almost perfect guess.
That is an amazing guess. You guys did amazing guessing what DNA could be. So let's move on to the next slide of what is DNA. So DNA is like the blueprint that we look at how our bodies develop and even some of our behaviors. Like when you build a house, you got to look at the blueprint on how to build the house. We receive half of this blueprint from our mother and the other half from our father.
So one of the main things we took away from that video is that DNA is made up of these different bases. These bases are the building blocks that makes up our genetic code. Just like Ariana and Zelda said, these are like the words in our recipe book that determines what our body looks like and does, and even somewhat how we behave. Instead of being made up of 26 letters, though, like our English alphabet, it's made of just 4 letters. So it seems like it should be really simple, right? The letters are A, T, G, and C. But with only these 4 letters, we can get all of the diversity and variety of different people and animals and plants and fungus and bacteria and everything alive in the whole world, just with 4 letters. G's never going to pair up with T or A, and C's never going to pair up with A or T. I like to remember this because G and C are both kind of curved. And so I know that those 2 letters go together. Yeah, great job, Luca. And then A and T are both kind of rigid. They both have straight lines. And so I remember the ones that with straight lines go together and the curved ones go together.
All right, guys. So that was so interesting. So the CRISPR-Cas9, what does it do? CRISPR-Cas9 is a genetic tool that helps scientists edit the instructions on the recipe book of life. You guys are all right. It does help edit DNA, so it can help cut, add, or even make it just stop from working at that moment. But it's still there, just deactivated. Imagine like a tiny pair of molecular scissors. It's cutting through. And these scissors help scientists use to edit existing genetic code or add new information to DNA. So just changing one little stripe of this can change everything on this DNA strand, which it makes CRISPR-Cas9 so exciting to work with.
Yeah, I want to talk a little bit more about how it was discovered. You asked about how DNA was first discovered. CRISPR-Cas9 was discovered in the 1980s that it existed. But they didn't realize it could be used as a genetic tool until 2012. So that was only 11 years ago. It was discovered by a female scientist, Dr. Jennifer Doudna. And this is a really cool story because Dr. Doudna just liked studying bacteria. She didn't think she was going to change the world. She didn't think she would find anything that exciting. She just loved bacteria and learning about them. So she was studying them, and she was getting some funding and doing her research and publishing, but no one really was paying attention to her. And then all of a sudden, she made the discovery that you could use this tool as a genetic tool to edit our own human DNA. And all of a sudden, everyone had all this attention, and she changed the whole world and is going to change your lives in many, 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 many ways. It's a really exciting tool because it can help us find cures for lots and lots of diseases and make amazing discoveries in genetics. And it has already been used to save millions of lives. Do you know how CRISPR was used to save millions of lives already just in 10 years?
I forgot what I was gonna say, but I also had a question.
Okay.
With what you were saying, did people even know that it was that existed, or they not until you discovered?
That's a great question. They discovered it existed in the 1980s, so not that long ago, only like 40 years ago. But they didn't understand what it could be used for until Jennifer Doudna discovered that in 2012. Juan, do you have a question, or do you have an answer to the question?
I actually have an answer to the question, how does CRISPR save lives? It saves lives because it's a kind of protein, and it protects us from viruses, and it protects us from getting diseases once we're a baby.
A really good guess. And then someday that's going to be true. Babies are going to be protected from disease with CRISPR, but that's not happening yet. That's going to be something that you as scientists are going to be able to work on and develop.
So please welcome our amazing guest, Miss Catherine.
Yay! Hi, Miss Catherine.
Hello.
Hi, everyone. All right. So, like Andrea said, I come from Senegal, Africa, and I moved all the way from Africa to UC Berkeley, in California, so that I can learn about how I can make our cells smarter and engineer them so that they can fight diseases, right? And to do that, I am using gene editing, right? Can anyone remind me what gene editing is? Let me try to see everybody. Neela? Is Neela?
Gene editing is when you take a part of your DNA out to change it and protect you from viruses.
Exactly. Anyone else? Arlen? Is it Arlen?
Gene editing is like from your DNA. If something happens to your DNA, it replaces it with a new one.
Exactly. So here I am trying to add something to the DNA so that our cells can become smarter and make new proteins and fight HIV, which is a virus, right? And HIV is a virus that infects people in Africa a lot, and that's where I come from. And so I want people in Africa to be healthier, right? That's why I came here, and now I'm doing a PhD at USC, which is right here in LA, and I'm studying gene therapy. So,
what does HIV stand for?
HIV stands for human immunodeficiency virus. So it basically infects humans only, and it makes your immune system weak. And your immune system is it's a system, it's a group of cells in your body that help you defend your body against diseases. So it protects you, it fights against infections like coronavirus and flu, and there's also cancer and other diseases. So HIV actually infects your immune cells, which are your defense cells. And how it does this, it goes to cells that are called CD4 cells. And when it enters in these cells, it basically cuts your DNA and it integrates its DNA inside your DNA, and then it stays in the body permanently. And what it does is it kills those immune cells that are supposed to defend us from diseases. when the HIV kills your immune cells, they can't fight the diseases. Like Juan said, you can get cancer, you can get other infections, you can throw up, you can just be sick and cough and not be able to do the everyday things that you need to do. Luckily, we have a medicine called antiretrovirals that can kill HIV or prevent it from integrating in our DNA, from spreading more in the cell, in the DNA, in the body and infecting other cells. And so these are medicines that most people that have HIV have access to. But the problem again is what Giovanna said. When the HIV gets in your brain, you can get sick, you can't learn, you can't talk, you can't walk sometimes. And so the problem with the anti-HIV medicine is that it can't get in the brain to attack HIV. So I use DNA to make these nanobodies. And then I add a structure in them, a structure that actually allows these nanobodies to go inside the brain, unlike the medicine that we currently have. And it's this green structure that also resembles a little bit those nanobodies from the alpacas and the llamas and the camels, right? And for it to get inside, you have these cells that are between your blood and your brain, and they form this barrier. They form like a big wall, or like a wall of soldiers, and they just prevent most things from getting in the brain, because you have to really protect your brain from almost everything. It prevents, that's why the medicine against HIV can't go in. But when I add this green little protein to the alpaca nanobody, then they can actually go inside the brain.